Welcome back, students. We are in the third video of our tissue systems in dicots and monocots. This is your chapter 6, Anatomy of Flowering Plants. So, we will move up towards the stem. Generally, stem is the aerial part of the flowering plants or a plant generally. So, in the anatomy of a dicot stem, what structures will we see and how these tissues are arranged, and this study are the main parts of a branch of biology which is known as anatomy. If it is this studying of internal structures of animals, there is anatomy in animals, but here we will see the anatomy of a dicot stem. So, in the ground plan of a dicot stem, what we will see that here the finger-like projections, tiny ones, they generally found in the yolk cells. This ground plan is basically of your primary stem, which has not undergone secondary growth. So what we will see here? In the ground plan, we will see that the epidermis is the outermost layer, and this epidermis bears epidermal hairs. And beyond epidermis, we will see hypodermis, which is part of the cortex. This is the cortex. Then this is the vascular system. Endodermis, the innermost layer of cortex. Then vascular bundles. So here is the important or interesting thing that the vascular bundles are arranged in a ring, or they are in a ring-like structures. And this is the reason why we will see secondary growth in a dicot plant. And the vascular bundles, here they are of open type, and here it is of endark type. That means the protozygotes are found towards the pith region, and metazygotes are found towards the periphery, towards the epidermal region. And they are conjoined type. What is conjoined? Both xylem and phloem are found in a single bundle. So these are known as the vascular bundles. And pith is well developed, and pith, vascular bundle, and pericycle, they together form a structure which is known as a stele. So, in the anatomy of the cord section of a dicot stem, what we will see, the epidermal hairs are present, and importantly here, cuticle encircles the epidermis. So cuticle is present here, and multicellular epidermal hairs, which are called as trichomes. They are called trichomes, are present. And sometimes these epidermal hairs, or the young stems, remember, this epidermis in some part it gets broken, or the structure which is known as stomata. Sometimes stomata are found in young stems, but generally stomata are found on leaves, but in young stems, when the leaves are not well developed, then the young stems bear stomata for the gaseous exchange and for the photosynthesis. So sometimes epidermis bear stomata also. So the epidermis is the outermost layer, and beyond epidermis we will see the cortex. But the cortex is composed of various types of simple permanent tissue, and the first one is a parenchymatous one, the next is chlorenchyma, and the parenchyma. So what is chlorenchyma? The chloroplast bearing parenchyma are known as chlorenchyma, and they help in they contain chloroplast. Sometimes in the young stem we are discussing, they help in the photosynthesis process. And the coleochromatous one, they provide mechanical strength. Then next is your endodermis. So endodermis is the innermost layer of your cortex, and endodermis here is rich in starch grains. And that's why these endodermal cells, which are rich in starch grains, they are known as starch sheath. So the endodermis, which is the innermost part of the cortex, bearing starch grains, are known as starch sheath. Then pericycle is present beyond the endodermis. And the endodermis is present beyond the phloem in a semilunar patches. So, like that, half circle, or we call as semilunar patches. So, these are the pericycles. So, these pericycles are present beyond endodermis and above the phloem as semilunar patches. And these patches are made up of sclerenchyma tissue. So if you remember, during the sclerenchyma tissue, the simple permanent tissue, we had seen that the sclerenchyma, they don't have any intercellular spaces in between these cells of sclerenchyma, and that's why they are very compactly arranged, which these sclerenchyma are found in the coconut, outer cell of coconut. So these sclerenchyma tissues composed or make the pericycle in a dicot stem. Then vascular bundles are arranged in a ring. So in the ground plan it is very evident that they are arranged in a ring-like structure. As it is a hot section, only two vascular bundles are shown here. Then in between the vascular bundles, so this is one vascular bundle and it is another vascular bundle. What we will see? We will see variously radially arranged cells are present there, and they are of parenchymatous one. So these structures, they are radially arranged and they are of parenchymatous cells. And these cells are known as what they are called? They are called medullary veins. So these medullary veins are present in between the vascular bundles and they are radially placed. The next one is a vascular bundle. We will see now how these vascular bundles, as we have studied, they are composed of a xylem, phloem. But here another one is present, which is a cambium, or it is also called vascular cambium. So this one is a phloem, this is a xylem. So xylem and phloem are present in a single vascular bundle. That's why the type of vascular bundle here known as conjoined. So they are present in a single strand. But in monocot root and dicot root, we have seen that they are present on different patches. But here they are present on a single vascular bundle, and in between xylem and phloem, the vascular cambium is present, and that's why this type of vascular bundle is known as open type vascular bundle. And next is a endartered form. So we have studied about exacts, but here it is endartered. So, but how it is different from exact? So in endartered type of protoxylem, it describes for that protoxylem or the fast formed xylems are present towards the pith region or towards the central region. So that's why it is endartered protoxylem, but the metaxylems are found towards the peripheral region. So here the vascular bundles are conjoined, that is both xylem not present on a single strand, and then they are open type, that is vascular cambium is present between xylem and phloem, and Next is entire protoxylem. Then we can see well developed pith is present. So this pith, muscular, strand and peristem. This all forms the still. So this is the general overview of the arrangement of different type of cells in a dicot root. So now we will see how these cells are arranged in a dicot stem. So if you want to write the answer of how the anatomical features or anatomy of epidermal cells, ground tissue system and vascular systems are arranged in a dicot stem, then first, as we have done in the last two classes, just start from your outermost layer epidermis. Just mention whether the hairs present are single cell or multicellular. As it is a dicot stem, it is a multicellular one. Then beyond epidermis we have the cortex, and this additionally hypodermis is present. And cholecylinders, pleurochylinders, parenchymatous cortex is present, and the endodermis here contains starch sheets. So the starch sheets are present in the endodermis, and xylem and phloem are present in a single gonad. That's why it is known as a conjoint. Cambium is present. That's why it is open type. And metaxylem are found towards the pith region. That's why it is endot type and pith is present. So this is the summary of the anatomical features of a dicot stem. So what questions you might be asked in the examination or in an entrance exams? This talks. What is conjoint type, vascular bundle, what are medullary rays, why the endodermis contains starch sheets. So all these questions prepare for this type. Just practice drawing these diagrams and start understanding the concepts. Then the studying the anatomical features will be very much interesting. So wish you good luck. Stay safe. Enjoy learning new things in your class 11 biology. Thank you. We will meet in the next class.